Hi again. Last week of the semester. That's exciting. So we're going to try and use this semester to wrap up a bunch of nerdy things and hopefully pull them all together in a context that shows you that so much of what we've been pushing towards all semester really does have biomedical relevance. So we're going to use things as diverse as Thevenin equivalent circuits to explain membrane electrophysiology, right? That's a big jump, but it's a jump I think we're ready to make. So we're going to break this into 3 videos, and this is the first in that sequence of 3. And each of them will, hopefully add another part, another perspective to, what it is we want to get across this time around. So this is about membranes and what's called electrochemical equilibrium. So equilibrium, that's a concept we've talked about throughout the semester. We're going to see another case of equilibrium here, and a very, very important one for how the body functions. And hopefully you'll see that, see some of that excitement in what I'll tell you about. So we're going to dive now into the area of electrophysiology, but also this is related to biomechanics. So it really does encompass both parts of the course, and the 2 organs that are most relevant for this are the brain and the heart, because, well, the brain not so much mechanical, there's not much movement going on in the brain, so we'll focus a little more on the heart, but with the knowledge that a great deal of what we say about electrophysiology will apply to both neurons and cardiac cells and skeletal muscle cells and smooth muscle cells and virtually every excitable cell that we have in our body. So it has a lot of relevance and breadth. The nice part about the heart, and the reason we've used it throughout the semester, is it has both mechanical and electrical aspects. And now we're going to move from that starting point of Wigger's diagrams and very high-level views of the heart. We're going to kind of dive right down beyond the cell to the cell membranes, actually, because that's where the story really all begins. And to bring you sort of back into that space again, and it is an again because we've watched this before, I'm going to give the floor to our friend Glenn Wilkenfeld, and Glenn will remind you about all the essential elements of membranes through this little video. So let's jump into the wrap video on membranes to hopefully remind you of some of your cell biology.
If you're in a cell cytoplasm heading outside, your last stop's a barrier 8 nanometers wide. The cell membrane, our subject for today, it's as basic to life as DNA. It's selectively permeable like a border patrol of a country. It maintains control, selecting what leaves or what gains entry. It's a guard, bouncer, watchman, patrolman, or sentry, but not just a guard. It sends signals and nerves. White blood cells use their membranes to eat germs like hors d'oeuvres. Membranes have receptors, enzymes, and junctions. So what kind of structure can have all these functions? C-double-L-M-B-R-A-N-E, control and transport selective permeability. Phospholipids, carbohydrates, proteins, and cholesterol, the fluid mosaic by layering us all. So let's take a look at what makes up the membrane. Phospholipids rule in this domain. In fact, membrane structure emerges directly from phospholipids' chemical properties. There's a head and a tail on every phospholipid. The tail's 2 long-chain fatty acids bound to a glycerol. It's made to order. The tail's non-polar, hydrophobic, fears water. The head's got a phosphate. It's charged negatively, makes the head hydrophilic, plays in water happily. So tail avoids water while the head's attracted to it. When phospholipids form the membrane, that's how they do it. Cause when phospholipids into water get submerged, a phospholipid bilayer structure will emerge. The tails hang together in a water-free zone. Hear the hydrophobic mode. Water, leave me alone. While the heads are sticking out, touching all those H2Os. Tails in, heads out. It's how every membrane goes. Tails in, heads out in a cellular sphere. It's the bilayered basis of membranes everywhere. C-double-L-M-B-R-A-N-E, control and transport selective permeability. Phospholipids, carbohydrates, proteins, and cholesterol, the fluid mosaic by layering us all. But a cell membrane's not just a phospholipid scene. There's cholesterol, carbohydrates, lots of proteins. In fact, membrane proteins have a presence so great they often exceed the phospholipids by weight. And all of these components are in constant motion, moving moving, mixing like a boiling potion, flowing like dancers in a party in Passaic. That's why the membrane's thought of as a fluid mosaic. Let's start with the proteins since they're key in this mix. Transmembrane proteins span the entire width. Typically they're ports, think of channels of conveyors for things that don't go through the lipid bilayer. Any protein embedded in the hydrophobic middle built right into the inside is considered integral, while peripheral proteins either hang on the exterior or on the cytoplasmic interior. Cholesterol keeps membranes flowing with ease. In cold it keeps the lipids moving so they don't freeze. In heat it slows the lipids down, limiting their traveling. Cholesterol keeps membranes from unraveling. Membrane carbohydrates work as markers or signs, so your immune system knows which flag your cells are flying. The blood types ABO and BNA are about the carbohydrates on red blood cell membranes. CLL-MBRANE control and transport selective permeability. Phospholipids, carbohydrates, proteins, and cholesterol, the fluid mosaic by layering us all. CLL-MBRANE control and transport selective permeability. Phospholipids, carbohydrates, proteins, and cholesterol, the fluid mosaic by layering us all.
All right, so what are the pieces of that fun little video that you have to really remember for our course? One of them is that ion channels are the resistors, if you will. They're the pathways for ions to move across a membrane. They're like our wires, if you will, but they have resistance. So they're like the resistors connecting the extracellular space and the intracellular space. So it's going to sound like a circuit and function like a circuit, and we can use some of the same rules as we use for circuits to analyze that behavior. So, and those ion channels, we call them, are really just proteins. They're folded up, chains of amino acids that are embedded into the membrane. So they break up the otherwise bilipid layer that you've just heard about, and are inserted into that bilipid layer, and the ones of interest to us at least span that entire bilipid and provide these pathways for ion channels to flow. And it's that behavior that we're going to try and capture in some of these equations and some of this circuit analysis. So again, we have a bilipid layer and we have ion channels that are embedded within it, and they're going to give us the behavior that we're really interested in. So they look something schematically like this, where you see the bilipids, and then embedded in that some proteins. Now each one of those proteins has different structure. You can see that in the schematic. It's also what we know is true in reality. And with that structure comes a function. And structure-function relationships are really at the core of physiology. This is what most of physiology is about. We've actually talked in terms of structure function at many different scales, and we're going to focus now on this membrane scale. So we have ion channels created through proteins, and each of those will have a structure, and that structure will determine its function. And we want to get a bit of a handle on how to describe that function. So there's some core ideas here that you've picked up from other courses, hopefully. We're going to focus on kind of the electrical aspects of just how we describe that ion channel movement, or that ion movement across channels, and really try and picture the cell as a circuit, or the membrane of the cell as a circuit. So there are lots of complexity to proteins and ion channels. There are whole courses dedicated to this. There are whole careers based on even a single subtype of an ion channel. So we can't get into that very much, other than to say that we have a transport mechanism that for ion channels is fundamentally passive. So remember we talked about active and passive transport. You'll have a whole course on transport coming up. So ion channels are really passive in the sense that they're just holes in the membrane. They're little tricky holes because they can open and shut. So they have these things called gates. But when the gate is open, then movement of ions through those channels happens because of those forces that we've already discussed. Those forces that involve things like concentration differences, charge, the electric field that is generated across that membrane. All these things come together, and that's really what we want to talk about in this lecture. So we have ions moving through membranes, moving across membranes through ion channels, and they're susceptible to all those transport forces that we talked about way back early in the semester. So we have spent a lot of time talking about electromechanical analogs. And you remember that we have notions of resistance in the mechanical and electrical side. We have notions of compliance, capacitance. We have a valve in mechanics. We have a diode in electricity. But now we're going to go to another set of analogs. So just like this set of analogs has been, I think, helpful for us to understand the behaviors we're trying to capture and to quantify those behaviors. Now we're going to discuss another type of analog, and these are the electrochemical analogs. So now we're going to bring two things together that don't necessarily seem like they fit that well. You've spent a whole year in Fund or a semester in Fundies 1 learning about the chemical approach to the body, and you spent this semester learning about the physics-based approach to the body. And now we're going to say they actually are in equilibrium of some sort. That should seem weird, and we'll explain how that works in just a sec. So on the electrical side, we have all these quantities that you've seen to some extent before, either here or in a physics course. The notion of electrostatic force, the notion of electrostatic potentials and voltage, a flow rate, which in the case of electricity is current. So all those terms exist, and you should have some comfort with those. It turns out they all have chemical analogs. And like other analogous quantities, there are similarities in function that we want to leverage as we decide on what sort of equilibrium might exist between them. So we have chemical force, for example, which has to do with the concentrations of those chemicals. We have the notion of chemical potential, the sort of potential energy of a chemical arrangement or chemical field. So that exists in chemistry just like it does in physics. And of course we have flow rates, the flux that you remember way back from Darcy's law when we talked about flux and that being a function of concentration differences. So this should sort of feel familiar. Even if it is a little bit weird. And in fact, this whole thing is so weird that how I like to use sort of, movie or story analogies, or metaphors, and I like this one a lot. I think it fits well. This is a movie now it's become quite old. 2014 it came out. Hopefully you can find it if you're interested. It's a fun, light sci-fi movie. I won't go into all its virtues, but what it does explain is this notion of parallel universes. And this is one way to think about the electrical universe and the chemical universe, that they're parallel in some way, and they're all sort of doing their thing in their parallel ways. And the question is, how do they ever interact? So if you want to get yourself in the right mindset, then check out Interstellar, which you can, of course, get through all the usual channels. So what does that parallel universe thing have to do with our situation? Well, here's an image of this sort of parallel universe that has, supposedly a wormhole, as the movie will explain, that connects the two universes. And it's not quite that metaphysical, of course, in our situation. There's a pretty clear connection between the two, but you can almost picture there's two universes and then try and say, how could they possibly be in equilibrium? How could there be movement back and forth between these two domains in a way that we can quantify and that makes some physical sense and is truly physics and not science fiction or metaphysics. And there's a simple way to get there, actually. And we're going to take a pretty classical path to this. There are other ways to think about it, of course, but this way I think is pretty robust. So picture, if you will, this thought experiment in which we have a chamber. And that chamber, glass, whatever, beaker, whatever you want to picture it as, has a membrane in between the two halves of that container. And the initial of that membrane we're going to picture as being impermeable, as in nothing goes across. So picture, like a thick piece of plastic, I don't know, whatever you want. So then we dump in some potassium chloride on one side into what was distilled water before. And then we see, okay, what have we done? What have we created here? Now we've created a diffusion gradient. If nothing else, we've created a diffusion gradient. Of course, we have no diffusion because that chunk of plastic in the middle doesn't let anything diffuse. So we have potential, we have concentration differences, we have the potential for flux, but we don't have the enabler for flux. So we have the drive, but not the enabler. And that should also ring a few bells from several times we've used that notion, that idea. We can also picture this membrane as sort of a very simple-minded, replacement for, and the membrane between the two halves as the membrane between the inside of a cell and the outside of the cell. So the whole time we're thinking through this, think the left side of this diagram is the inside of the cell, the right side is the outside of the cell. And so we have a chemical gradient, even though, again, we have no enabling and hence no flux. Now, like magic, we're going to turn that membrane into something that's semi-permeable, which means it lets certain ions through and other ions it doesn't let through. So what happens now? So it's semi-permeable in a very special way in that it allows potassium ions to get through. Don't worry about the magic of this quite at the moment. It turns out ion channels have this magic, but just bear with me. So we have a semi-permeable membrane that allows potassium to flow through. Now we have the enabler. So we have the force, the driving force, we have the concentration gradient, we have the enabler, flux will happen. With that flux, of course, because these are charged ions, ions have charges, and this has a positive charge, as every ion moves from the left side to the right down its chemical concentration gradient, it will take charge with it. And when charge moves in space like this, then it creates a potential difference. We've learned about voltages and potential differences. So now we have a situation where we have diffusion pushing potassium, in this case from left to right. Now, however, as the charge builds up, as the charge separation occurs, then we're going to have more and more positive charge entering the right-hand side of the chamber, and eventually that positive charge will build up enough that it will sort of push back the entry of more potassium ions, positively charged potassium ions. And you can already imagine we're heading towards some kind of equilibrium here that balances the diffusion gradient that we have that's chemical and the electrostatic force or the electrostatic gradient, which is physics, which is electrical. So we're going to bring those two at some point into balance. Now in this diagram, we've showed, I've illustrated that with some arrows. The arrows are meant to represent the dark arrow. The black arrow is meant to represent the chemical gradient. The open arrow is meant to represent the electrical gradient. And initially, we only have chemical gradient. And then when we become semi-permeable, the chemical gradient exceeds the electrical gradient. But you can imagine that over time, that charge will build up to the point where no more ions of positive charge can actually move to the right because the voltage has built up or because the electrostatic repulsion has built up so much on that right-hand side that it's pushing those back. And we get a situation of no net flux. That should sound like equilibrium, or at least one form of equilibrium, and indeed it is. And that's the third stage of this process, where we have an equilibrium and no net flux. So the big question here to think about is what exactly is in equilibrium? Whenever we have equilibrium, our first question should be what's balancing what here? It doesn't mean, for example, the same as if I'd said, I'm going to put potassium ions in the corner of this beaker and let it diffuse throughout the beaker. Under those conditions, we would see, of course, a uniform distribution of those potassium ions. They would keep moving until they were uniformly distributed throughout the space. That's not what we have here. Because we have this semi-permeable membrane, because every time a charge moves across, it creates a voltage difference or a difference in electric field, that's a different scenario. So that semi-permeable membrane brings about some really important changes because we have to have charge balance. And in this semi-permeable membrane, the chloride ions are not allowed to move. They're stuck on the left-hand side, so they're bringing in the negative charge there. We have positive ions leaving, and it's creating this difference in electron charge concentration and hence a force that will push ions from the right back to the left. At the top of this figure, you can also see there's a little voltmeter going back and forth. And that reflects a true voltage difference that will arise between the inside and the outside of a cell or on either side of a semi-permeable membrane. And that voltage is really one way to measure our equilibrium, because there will be a voltage which is strong enough to perfectly balance the concentration difference that we have between the left and the right-hand side. And of course, that equilibrium voltage will change if I change the concentration. If I now at this point add in a little more potassium on the left-hand side, I'll increase the concentration gradient. That will push even more ions to the right because that diffusion gradient, that solid air, will become a bit longer until there's a new equilibrium. And in that new equilibrium, the electrostatic gradient pushing from right to left, the open arrow, will also become bigger. And at that point, that voltage will have tipped even further over and become larger. So that equilibrium voltage then becomes a metric for the diffusion gradient that we have across that semi-permeable membrane. So they're completely coupled. So with diffusion gradient comes electrostatic potential that balances the two at equilibrium. And that voltage, that specific voltage, is called the Nernst potential. It is the equilibrium voltage, equilibrium potential that exists, and it depends on the concentration difference. Again, the more concentration difference I have, the larger that voltage has to become so that equilibrium can occur. So equilibrium voltage is not zero. It's definitely not zero. You can also imagine that because positive charges have left the left-hand side and gone to the right, the left-hand side becomes more negative compared to the right-hand side, right? There's a net movement of positive charge to the right, so the right-hand side will become more positive. And if that inside-outside explanation holds true, then I will say that the inside of the cell becomes more negative compared to the outside of the cell. And that is indeed what happens in real cells. So that's the concept of equilibrium potential. And it's incredibly powerful, incredibly important. It's also very specific to certain ions. Each ion will have its own equilibrium potential, and they'll act independently. So in this case, potassium has an equilibrium potential. You can imagine chloride might. Chloride's a little bit different. We'll come back to that. But potassium will, sodium will, calcium will, all the ions that we sort of see in living systems or living cells, they'll all have their own equilibrium potential, which again will be a function of their concentration and their concentration gradient. And now if you look in the textbook, you'll see there's a nice equation, pretty nice equation. I don't know, how do you determine how nice an equation is? It's a nice equation. It has a log in it, so it's got a little bit of complexity, but it's a very nice equation because it really describes a lot. It tells us quantitatively, if we have this concentration difference, we have this equilibrium potential. So that's the wormhole that allows us to connect the universe of chemistry with the universe of electricity, and that's what plays out in our cells, in every one of our excitable cells. Every day, every minute, every second. So again, there are some characteristics of this equilibrium that are worth noting. There's kind of three major ones to keep in mind. It's concentration driven, concentration gradient driven. It's separate for each ion. When we have equilibrium, it does not mean the concentration is equal. It does not mean the voltage is zero or equal. It means that there's no net flux of ions. That's how we define this particular equilibrium. And finally, once the equilibrium is established, it is there as a passive process. Nowhere in this process did I say there was a battery or that there was an energy source that had to be put in place. The semi-permeable membrane seems magic, but that's a passive element as well. So this whole thing plays out without energy input. So it's a purely passive situation that will cause ions to flow across the membrane and will create a battery, will create a voltage difference across the membrane. And our next step will be to figure out how do we build a circuit around that type of thinking about a membrane, given that there are going to be multiple ions and even some capacitance. So that's the next video. Have a look at that one next. Hope this was helpful. See you soon.